Okay, good morning everyone. So in the second part of this talk I'd like to give a brief update on the frost pre-breeding research that we're doing at the University of Adelaide. So there's two broad aims to our research projects that we have. The first is to continue to develop methods to reliably screen for frost tolerance in the field. And then the second is to really use these methods to identify new sources of barley and wheat germplasm for use in breeding programs that offer better tolerance. So to develop the methods, we've used several sites around South Australia in the coldest cropping regions, and the sites here are shown with the red dots. The site that we mainly use now, and we've put a lot of resources into developing a frost screening nursery, is at Loxton. There's a couple of reasons for that. One, it gets reliable spring frost events. And the second, it's located next to the Murray River, so we've got access to irrigation water to help establish our trials. So this graph here shows the number of days per month below zero at our Loxton site based on historical data. So we sow our trials to try and get them to flower during this window between mid-July to mid-September, so that we have an increased probability of getting data from our trials. So to target that window, we use around 7 to 8 times the sowing, and they're spaced between April to June. At our site, we have a weather station installed and temperature sensor probes throughout the trial so that we can measure the temperature at the canopy height. It's worthwhile noting that actually at our site is the Bureau of Meteorology weather station, which is actually about 20 metres away from our trial. And the temperature there is measured at a slightly higher level than what the canopy height is, and we see quite a difference in the minimum temperature readings from these two sources. So we might get say a +1 degree temperature from the bomb site, but actually at canopy height it might be -2, and we start to see frost damage within those trials. So that's something to be mindful of. So we've also spent a lot of time developing standardised phenotyping protocols. So when there's a frost event, we'll go out and specifically tag those heads that are at the correct developmental stage at the flowering point. And then a couple of weeks after that, we'll go out and collect those heads and count the number of sterile grains. And that gives us our score, which we refer to as % frost-induced sterility. So it's quite labour intensive, but we can get through quite a number of samples per year and analyse a large number of genotypes. So this is what the typical sort of damage that we see in our trials. So in barley it's quite obvious. You can see I think it's pointed to work. So you can see sort of the random sterile florets within the head caused by the frost. In barley it's a bit more tricky. Often you won't see any damage at all, unless it's a really severe frost, and then you'll start to see some bleaching of the heads. But yeah, often in wheat there's no visible signs of damage. So some of the early work that we did was in barley, and one of the major outcomes of that was the identification of some improved tolerance from the Japanese varieties, namely Haruna Nijo and Marji Nijo. And an outcome of this was that breeding programs then adopted these varieties and started transferring the tolerance into adapted South Australian varieties and breeding programs. So over several years now we've been analysing the effectiveness of the Japanese derived tolerance, and this graph here shows data from 3 years. And you can see this on the left axis here we have the frost-induced sterility. So the smaller the bars, the better, the less damage. On the left here, you can see that we've got the Japanese varieties here, the Marji Nijo and Haruna Nijo, showing good levels of tolerance. We've also got an adapted breeding line here that contains the frost tolerance genes. And what we also found is that Kiel was actually performed really well in terms of frost tolerance over our long-term trials. But the other point to take from this is there is a large extent of genetic variation for frost tolerance within barley. So we've also been doing these trials in wheat, trying to identify new sources of frost tolerance. And this is from a national trial that we've been running. And here we have 3 varieties, kite, young and wild catching. And the data here is shown from 3 years and 10 separate frost events. And on the bottom here we have the minimum temperature for each of these frost events. So you can see again there is consistent levels of genetic variation, with kite and young offering or showing better levels of tolerance, and wild catching generally having higher levels of damage. But the important thing to note here is that there's a threshold of about -4 degrees, so if the temperature goes below that, then all varieties are pretty much equally susceptible. So this is some data that we've got from Ben Bidoff at DAFWA, who's also part of this national project. And in the first 2 years, his trials were pretty much knocked out by drought, but in the final year here, we were getting consistent rankings between the 2 states. So we've also been looking at larger germplasm sets in wheat. So this is some data from our 2010 trial. So the point to note here is that there is quite extensive genetic variation for frost tolerance. And the rankings that we got were generally consistent. So you can see here we have young kite and wild catching. One thing to note here is that we do have this one line, OZ 30323, which showed quite good levels of frost tolerance. And we're now working quite a bit with this line to try and understand the genetics of that. And this line's also been now given into breeding programs so they can start incorporating that. So how does frost affect yield? That's one question that we've really had over a number of years now. And a couple of years ago there was an AGT yield trial that got affected by frost. So it gave us a good opportunity to go out and measure the damage within that. And looking at the mean of this trial, we can basically say that for each percentage increase in frost-induced sterility, generally reduces yield by about 1%. Now saying that, we really need to look at this in more detail and see how different varieties vary in regard to this, and also across different environments. So the take-home messages from this presentation really are that reductions in grain number due to frost started at about 0 degrees in wheat and -2 degrees in barley. And that you might not actually see any visible damage in the crop, especially in wheat. Large losses in grain number start to occur at about -2 degrees in wheat and -6 degrees in barley. And from the pre-breeding research we've been doing, we've identified improved genetic sources of frost tolerance for both wheat and barley, and these are now entering into breeding programs. So lastly, I'd like to introduce a new project which is starting this year. It's called the Australian National Frost Project, and the main aim of this is to start to develop frost sensitivity ratings for the current wheat and barley varieties, so that farmers can begin to use this information in variety selection. So it's going to be using Australian-wide trials based at Loxton in South Australia, Merredin in WA and Narrabri, New South Wales. Initially it's a 5-year project, and we're going to start to provide the ratings after the third year of trials. So I'll wrap it up there, and thank you for your attention.
Tim, Western Australians claim that yip pea is relatively frost tolerant. How does that line up with your experience?
I guess we could say from our trials so far that it's sort of mid-range. I wouldn't say it's spectacular, we wouldn't say it's excellent. But again, yeah, I mean, it's going to really come down to running these trials over several years and at the different sites before we can really be confident in making any claims about that.
How much of that frost tolerance is actually related to flowering time? So are some varieties just avoiding frost? Is that why you're getting a higher rating?
So the reason I showed some of that national data is that those 3 lines actually flower pretty much at the same time in our trials. So at least in that data set we can remove maturity. But across the larger data sets, it could come into play. But to get around that, really our tagging method of only tagging at tagging at flowering when there's a frost event should hopefully combat some of those problems as well.
Tim, have we got any understanding of the mechanisms of frost tolerance or avoidance, and have you identified any genes that are related to that?
Yeah, so something I didn't speak about today is a lot of the genetics work that we're doing. So in particular, we're doing some genetic studies looking at wild catching and the tolerant or the susceptibility mechanisms in that. And likewise, we're also working with this OS30323 line and looking at what's the tolerance mechanisms in that line. I've got some good initial results. It looks like it's related, the mechanisms are related between wheat and barley. So we're working further in that regard.